Hello, I'm Dr. Andy Groves from Baylor College of Medicine in Houston. I'm a member of the Hearing Restoration Project that is organized and funded by the Hearing Health Foundation. And I'm here today at the offices of the Hearing Health Foundation to introduce you to the Hearing Restoration Project and to tell you a little bit about the science and the scientists that are behind this new venture. So let's begin at the beginning. As I'm speaking to you now and you're listening to the sound of my voice, sound waves are traveling across the room and are being captured by your external ear. The sound is then transmitted from the external ear across the middle ear space by the three tiniest bones in our body, and these three tiny bones impact against the inner ear that creates waves in liquid that our inner ear perceives as sound. Now, as I'm sure many of you know, the business end of hearing is a structure called the cochlea in the inner ear. And what I'm doing here is illustrating the inner ear in a newborn mouse baby. And I'm illustrating the cochlea, the business end of hearing, with this yellow arrow. If we look inside the cochlea and cut a slice through it, the business end of the cochlea that actually does the hearing is a structure called the organ of Corti. And the organ of Corti contains tiny sensory hair cells and their surrounding supporting cells that are responsible for perceiving sound. If we cut the top off the organ of Corti in this cartoon and look down on it from above, you can now see the arrangement of hair cells and supporting cells viewed from above. And you can see at high power the tiny microscopic bundles of hair that project up from the surface of each hair cell. And here on the right, I'm showing you the surface of a single hair cell. And you can see the hair bundles sticking up from its surface. And sensory hair cells are sound receptors that are activated by mechanical force. So the sound waves that are passed along the cochlea cause these bundles to be deflected, and the deflection of these bundles makes the hair cell become electrically active, and a nerve cell then connects that hair cell to the brain, where our brain perceives the electrical signals as sound. Now, this is a wonderful system, and it allows us to hear speech and conversation and music, and we can hear extremely quiet sounds. Now, the problem with a structure that is extremely mechanically sensitive is it's very easy to damage it. And over here on the right, I'm showing you an organ of Corti that has been damaged by intense sound. And I think you will appreciate that many of the hair cells have been lost from this organ of Corti, and as a result, the animal would have a hearing loss. Now, there are many things that can kill or damage hair cells, and I've listed a few of them here. They will be familiar to you: explosions, heavy machinery, gunfire, rock music, and iPods, and so on and so forth. In addition, there are certain drugs, in particular chemotherapy drugs, and particular kinds of antibiotics that are also very good at killing hair cells. Finally, if you are fortunate enough to avoid the hazards on the left and stay away from the drugs on the right, we know that old age is also very good at killing hair cells. And in fact, about 50% of the population, by the time they reach retirement age, report some form of hearing loss that is typically due to the loss of sensory hair cells. Now, that's the bad news. But the good news is that many vertebrates are capable of regenerating their sensory hair cells. And in fact, fish and frogs and birds and reptiles can all perform hair cell regeneration. What I'm showing you here are some slides from my colleague in the Hearing Restoration Project, Ed Rubel. And this is looking at the surface of the hearing organ of a bird. And if you then damage this hearing organ and look at the organ after damage, you can see that many of the hair cells have been destroyed and are missing. However, over a matter of weeks, new hair cells appear, and you can see them illustrated here, in this case with some arrows. And these new hair cells, after they have grown back, are capable of participating in hearing. And indeed, birds can recover their hearing almost perfectly after being damaged by sound or chemicals. Now, it's been known for over 20 years that birds are capable of regenerating their hair cells. And we now know quite a bit about the mechanism by which birds can do this. So I'm illustrating this in the form of a cartoon, showing you the time course of damage and regeneration going from left to right. So on the left, I'm showing you hair cells in purple, and they are surrounded by other kinds of cells called supporting cells. And after damage, when the hair cells are killed, some of the supporting cells are induced to divide, and I'm showing this in yellow here. And when a single supporting cell divides to make two daughters, one of those cells remains as a supporting cell, and the other cell turns back into a hair cell. And so you can see by the time we get over to the right-hand side of the screen, you can see that after regeneration, the system has been restored back to normal, with hair cells and supporting cells being able to participate normally in hearing. And it's possible to take a bird and to damage and kill its hair cells several times, and after each injury, the supporting cells are able to start dividing and will give rise to hair cells again. Now, in fact, this happens, this process of regeneration happens in many parts of our bodies. For example, in our skin is constantly being replaced by the division and differentiation of cell types. Similarly, in our digestive system or our bone marrow. However, in our hearing organs, in our inner ear, this form of regeneration never happens. And as a result, in mammals in general, and humans in particular, hearing loss is progressive and permanent. Now, mammals do have hair cells and supporting cells in their inner ears. However, after hair cells are killed in mammals, the supporting cells do not divide and they don't turn back into hair cells. And this could be for several reasons. It might be that mammals lack the capacity for regeneration. Alternatively, it is possible that mammalian cells do have the capacity for regeneration in the inner ear, but that the regenerative signal that starts the process is missing. Another possibility is that these cells receive the signal, but the pathway inside the cell that allows regeneration to happen is blocked. So we can think of the challenge of regeneration as a simple formula. And I've illustrated this using the bird as an example here. Put simply, regeneration is simply supporting cell division and then differentiation into hair cells. And if we can understand these two processes, it may be possible to bring about regeneration in mammals and ultimately in humans. So the Hearing Restoration Project has set a goal for the first 5 years to start to try and understand why it is that mammals are not able to regenerate their hearing cells, whereas other vertebrates, such as fish and birds, are able to do so. Briefly put, we know that birds and fish are readily able to regenerate their hair cells. Members of the Hearing Restoration Project in the past have shown that, in fact, in newborn mice, it is possible to generate new hair cells in special experimental circumstances. However, we also know that mature mice are unable to regenerate their hair cells. And what we hope to do in the first 5 years of this project is to analyze genetic pathways that are occurring as regeneration happens in fish and in birds, and to try and understand why these pathways are not active in the mature mammal. Now, some of you might be asking, why are we looking at fish and birds and comparing them to humans, where fish and birds separated from us in evolutionary time about 450 million years ago? And in the next few slides, I want to try and provide a little justification as to why we think it's important to focus on animals that can regenerate and compare them to animals that cannot. Now remember, when we're understanding the genes that are used during regeneration, genes are simply DNA building blocks. So DNA is a long molecule that is present in every one of our cells in our bodies. And DNA is simply a series of blocks, we call them C and T and G and A, that are arranged over and over again in every cell. And a gene, whether it is a human gene or a mouse gene or a fish gene, is nothing more than a sequence of these chemical building blocks. But the basic principles underlying what makes a gene and what the gene does are the same, whichever animal you're looking at. To give you an example of this, let's think about languages, which are also a series of building blocks. So if we take the English phrase good morning and we compare it to the same phrase in other languages, what I hope you can appreciate is that these 5 other languages, who all have a phrase for good morning, the arrangement of building blocks, or letters in this case, is actually very similar to the English, although they have diverged slightly. Nevertheless, even though the words have diverged slightly in their sequence, they carry the same function. So all these phrases mean the same thing in English or in Danish or in Norwegian. And similarly, in different species, even though the chemical composition of the gene might be slightly different, the function of that gene is identical. Similarly, we know that animals have evolved over millions and millions of years. And just as words can change over evolutionary time, so genes can gradually change their sequence too. Now, to illustrate this, I have taken a phrase that has been used again and again over the last 1,000 years, and I'm showing you how this phrase changes over evolutionary time. Now, I'm sure you don't understand at all what this phrase says, but it was nevertheless spoken quite a lot by Anglo-Saxons in the 10th century. If we now move forward 200 years and see how the same sentence has evolved, if you look closely, you might be able to see things in it that are familiar to you. If we move forward another 200 years, you can start to see that there is definitely a recognizable pattern here. If we move forward another 200 years or to the present day, you can see the meaning or the modern day meaning of this phrase. But if you trace it back, you can see that the individual letters that make up this phrase have changed over time, but the meaning remains the same. Similarly, the DNA blocks of genes in related species have changed over millions and millions of years, but frequently keep the same function. Now, to give you a really striking example of how identical genes can have very similar functions in different species, I'm illustrating this to you with a particular gene called c-KIT. Now, what you're looking at here on the left is a baby that has a mutation in its c-KIT gene. And you'll notice that the baby has a region of unpigmented skin in the center of its forehead. Now what I'm going to show you is a mouse that carries a mutation in the mouse version of this gene. And you can see that just as the baby, the mouse also has an unpigmented patch of skin in the center of its forehead. The point of this is that the baby and the mouse each have a c-KIT gene. The gene has the same function, even though its sequence is slightly different in the two species. What we hope to do by looking at the genes that are responsible for regeneration of hair cells in birds and fish is to compare these genes to mice and eventually humans to understand why mice and humans cannot regenerate their hair cells. So really what we're looking for is the genetic pathways that regulate regeneration. Genes don't work by themselves. They tend to work in groups or in pathways. And we believe it's extremely unlikely that we will discover a single gene that will be a magic bullet that would trigger regeneration if we somehow knew how to switch it on. Rather, what we hope is to discover sets of genes that act together in pathways to regulate behaviors such as cell division or cell differentiation. Now, I want to illustrate to you this idea of genetic pathways. And since I'm talking to you today from Manhattan, I thought it would be a useful analogy to tell you about the island of Manhattan, which is about 33 square miles, and it has a population of over 1.5 million people. Now, Manhattan has an enormously complicated infrastructure that is vital for the city to function. And indeed, many aspects of its infrastructure are common to all cities. It has a water system, it has a power system, it has a sewage system, a transport system, a law enforcement system, and so on. And by way of illustration, I'm illustrating the New York subway system here. This is vital for the city to run. Now, in contrast, let's think about Los Angeles, also a major American city. However, this city is over 500 square miles and has a population of nearly 4 million people. And although it has many things in common with Los Angeles, with Manhattan, it has a power system, it has a sewage system, it has a law enforcement system, it also has fundamental differences. And by way of illustration here, I'm showing you the public transportation system of Los Angeles, which you can see is nowhere near as comprehensive as it is in Manhattan. And in fact, LA had a reasonably good transport system until the 1950s, but it gradually degenerated and has been rebuilt only very slowly. The point I'm trying to make here is that if you think of New York and Los Angeles as different species, their cells have many things in common. However, there can be many important differences between them. And what we hope to understand is the pathways that underlie aspects of regeneration, like cell division, to see what the differences are between different species. The way we're going to do this is by comparing the genes that are switched on during regeneration in birds and fish, and compare these same genes and to see what happens to them when we look at failed attempts at regeneration in mice. And we're going to examine the genes that are being switched on or switched off in these two systems using modern DNA sequencing technology. And this allows us to read out the chemical components of huge amounts of DNA. So as a cell undergoes changes, it switches on different genes, and we will be able to read which genes are being switched on or off after damage or during the process of regeneration. Now, in the last few years, advances both in bioinformatics and sequencing technology have allowed us to perform these readings of chemical building blocks on a massive scale. And again, by way of illustration, when I first started my graduate studies in 1988, it would take a scientist working flat out maybe 3 years to just read 8,000 building blocks of DNA. Nowadays, it's possible for the same scientist to read over 10 billion building blocks of DNA information in a working week, in just 5 days. And so the rapid advance in technology has allowed us to analyze genetic changes that are occurring very, very quickly and very efficiently. And what we are going to do is apply this technology to look at genetic pathways that regulate regeneration in birds and fish, and to see what is happening to these pathways in mammals. And so what I'm going to do in the rest of the talk is to illustrate 2 examples of genetic pathways that may regulate hair cell regeneration. So the first one is the first piece of this equation, which is what makes supporting cells divide. So some time ago, my colleague Neil Siegel and I, who is also a member of the Hearing Restoration Project, tried to answer this question in mice. And we simply wanted to ask, are there any circumstances under which mouse supporting cells can divide? So to do this, we made a genetically engineered mouse in which we labeled the supporting cells with a fluorescent green protein. Now, this green protein is normally made by jellyfish, but through genetic engineering, we were able to splice this gene and put it in the mouse so that it would be switched on in supporting cells. We developed ways of purifying these supporting cells, and we developed ways of growing the supporting cells in a dish to ask, are these supporting cells able to divide, and can they make hair cells? And when we did this, we found that supporting cells are indeed able to divide. To do this, we had to feed the cells with a substance called epidermal growth factor, or EGF. And when we give this factor to supporting cells, they can undergo division. And I'm showing this to you here by feeding the cells a compound that makes the cells go red when they divide. And so you can see that in the center here, there are plenty of supporting cells that are dividing. And indeed, some of them go on to make hair cells, which is shown by the green marker here. Now, what EGF is doing by making these cells divide, we found out, is it's switching off another gene called P27. P27 is a gene that blocks cells from dividing. So what I've done here is to show you on the left a group of cells, and I'm labeling the dividing cells in red. In the middle panel, I'm showing you the same cells, but in green, I'm showing you the cells that are making P27. And so when I put them together on the right, you can see that all the cells that are dividing have switched off P27. There are no cells here that are both green and red. And so what we think is happening is that EGF is switching off the P27 gene and allowing the supporting cells to divide. And in fact, we have demonstrated this further by using drugs that can block the EGF pathway. And I'm showing you these drugs in the center panel and on the right-hand panel. And the bottom line in this slightly complicated slide is that basically if we switch off the EGF receptor pathway, the cells cannot divide and they cannot switch off P27. Now, interestingly, this process also seems to happen to some extent in birds. So as I told you some time ago, we know if you kill bird hair cells, the supporting cells surrounding them start dividing. And I'm showing this to you here. On the left-hand side, you're looking at the surface of a bird hearing organ, and all the hair cells are shown in green. On the right, I'm showing you a hearing organ where all the hair cells have been killed, and now the dividing cells are shown in red. And you can see that after killing the hair cells in a bird, many of the supporting cells start dividing. However, if we now block the EGF signaling pathway in the bird, we can significantly reduce the number of dividing cells. So if you compare the upper two panels that show you the division in a bird after damage, and the lower two panels show you a similar experiment where we have now blocked the EGF signaling pathway, it should be clear to you that we've significantly reduced the number of dividing cells. So these experiments suggest that the EGF signaling pathway is one of the pathways that supporting cells use to decide whether they're going to divide, and that this pathway is used both in birds and mammals. I think this offers a good illustration of the fact that there are genetic pathways that exist in many different kinds of vertebrates that may be shared in processes such as regeneration. And what we have to understand is why EGF cannot trigger regeneration in mammals, whereas it can do this in birds. And I should add that this work was done by 3 members of the HRP consortium, myself, Neil Siegel in Los Angeles, and Jennifer Stone at the University of Washington in Seattle. Our hope is that if we can compare normal and regenerating fish and normal and regenerating birds with young mice in which we're able to stimulate regeneration in certain circumstances with undamaged mice, and compared these pathways with mature mammals that can't regenerate, we can start to find some of the similarities and differences that may underlie the decision for cells to divide and participate in regeneration. And a number of the scientists in the Hearing Restoration Project are collaborating together to work on different aspects of these projects. Now, I just want to emphasize why it is important to compare young mice with older mice. So if you recall, I told you that in certain circumstances, the supporting cells of young mice around the time of birth are able to divide and give rise to hair cells, just like what we see in birds. However, mice don't actually start to hear until they're about 2 weeks old. So unlike humans, mice don't hear inside the mother. They only start to hear when they're about 2 weeks old. And prior to this, the organ of Corti is not fully mature. So when we went back and repeated our experiments on mouse supporting cells, but did them in 2-week-old mice, we found a rather different result. Instead of the supporting cells being able to divide, what we found that in 2-week-old animals, almost none of the supporting cells were able to divide, and none of them were able to switch off the P27 gene. So something was happening between the time of birth and the time a mouse is 14 days old that causes the supporting cells to change their behavior. Now when we give them epidermal growth factor, they're unable to switch off P27 and they're unable to divide. However, we do know that P27 is at least partly responsible for this change in supporting cell division, because if we use mice in which we have genetically inactivated P27, what we find is that more of the cells are able to divide. Instead of less than 5%, we now have closer to 15% of the cells that can divide when we genetically inactivate P27. And what this tells us is that P27 is one of the genetic components that underlies this change in behavior. And so this, I hope, can illustrate to you why it's important to compare young and old mice, because we observe some aspects of regenerative behavior in young mice, but in a relatively short period of time, we find that this regenerative ability is lost. And so by comparing young and old mice, together with comparing mice and birds and fish, we hope to understand some of the genetic pathways that may be responsible for causing cells to divide during regeneration. Now what I want to do is turn to a second example in the last part of this presentation. So if you recall, I told you that the formula for regeneration must involve supporting cells dividing and supporting cells turning into hair cells. And so in the second example, I want to talk briefly about ways in which we can make more hair cells and the genetic pathways that might underlie this process. As you can see here, after damage to hair cells, the organ of Corti has lost many of its hair cells, and only in some regions only the supporting cells are left. And so in order to try and enhance the production of hair cells, we want to understand pathways that might make supporting cells turn into hair cells. Now, for many years, people have suggested that in the organ of Corti, there might be an ongoing conversation between hair cells and supporting cells, such that hair cells actively tell the supporting cells to remain as supporting cells and not to turn into hair cells. And as a result of this ongoing conversation, you would have an unvarying mosaic of hair cells and supporting cells in a regular and repeating pattern. And I'm illustrating this on the left by showing you a bird hearing organ, and on the right by showing you a mammalian hearing organ. And I hope you can appreciate that there is a repeating and very fine-grained mosaic of hair cells and supporting cells in each case. And in fact, if you look across the animal kingdom, you can find many aspects of repeating cellular mosaics. On the left-hand side here, I'm showing you an insect compound eye containing over 600 individual lens-bearing eye units that are arranged in a mosaic pattern. On the right, I'm showing you the mosaic pattern of sensory bristles on the surface of an insect. And in both cases, these patterns, these cellular mosaics, are established by a genetic signaling pathway called the notch signaling pathway. Now, the details of this are not important. The bottom line is that notch signaling is a molecular mechanism that cells use to make fine-grained patterns of one cell type and another in developing tissues. And in fact, what we know is that the notch signaling pathway is active in the inner ear. And in fact, hair cells signal to supporting cells through the notch signaling pathway and actively prevent them from turning into hair cells. The prediction would be that if you can block this conversation, if you can block the notch signal that is being delivered to supporting cells, it might turn those supporting cells into hair cells. Now, in fact, it is possible to do this. So my colleagues and I have developed a simple system to investigate this, in which we can take a newborn mouse cochlea, and here we have used genetic engineering technology to make the hair cells fluorescent green. And we can grow this cochlea in a culture dish, and we can expose it to drugs that inhibit the notch signaling pathway. And what you see here on the left-hand side is a normal mouse cochlea that's been cultured for a few days just by itself. And you can see that it still has plenty of hair cells. However, if we block the notch signaling pathway, as you can see on the right here, you can see that there are many, many more hair cells. In fact, we see a 50% increase in hair cells when we grow these cochleas in drugs that block the notch signaling pathway. In fact, the supporting cells are turning into hair cells. This process also happens in other vertebrates. For example, in fish. Fish have hair cells all over their bodies that allow them to detect fluid movement in the water. What I'm showing you here is the work from 3 members of the Hearing Restoration Project, Tatiana Piotrowski, Dave Rabel, and Ed Rubel, who will work on zebrafish as an animal system. And in these 3 pictures, you can see patches of hair cells in fish by these bright green dots along the body of the animal. So in the top, I'm showing you a normal zebrafish larva. In the next panel down, I'm showing you a fish that has been exposed to antibiotics that kill hair cells. And you can see that after 24 hours, some of those patches of hair cells are now starting to come back. And this is because the supporting cells are turning back into hair cells. And we know that this process is regulated in part by the notch signaling pathway. Now, I just want to play you a short movie to show you this happening in real time. One of the beautiful things about zebrafish is that we can observe their hair cells dying and regenerating in real time. And in this time-lapse movie, I'm showing you one sensory organ on the side of a zebrafish, in which the hair cells are shown in green. And we have killed many of the hair cells here. But as you can see, over time, and the time here is shown in minutes and hours, you can see new green cells that start to appear in this sensory organ in the fish. So up to now we have about 6 hair cells present in this organ. At the beginning there were 2. And I think we're going to see 2 more cells generated in another short while. So by the time this movie comes to an end, there are now 8 hair cells, where at the beginning there were 2. This process took just 22 hours to regenerate 6 sensory hair cells. And this process is regulated by the notch signaling pathway, just as it is in mammals. So on the left here, I'm showing you a normal regenerating sensory organ in fish. And on the right, I'm showing you a similar sensory organ that has been treated with drugs that block the notch signaling pathway. And what I hope you can appreciate is that there are many more hair cells on the right than on the left. So once again, notch signaling also works to regulate the proportion of hair cells and supporting cells in fish, as well as in mice. And in fact, this is also true in birds as well. So here is a hearing organ in birds that has been damaged and is starting to produce more hair cells. And if we take a damaged bird hearing organ and grow it with drugs that block the notch signaling pathway, I hope you can appreciate that there are many, many more of these red hair cells in the drug-treated organ compared to an organ that hasn't been treated with drug. And once again, just as in birds and fish and in mammals, in each case, blocking the notch signaling pathway allows us to generate more hair cells. And so what this suggests is that many vertebrates, fish, birds, and mammals, all use this genetic pathway, the notch signaling pathway, to decide whether to become a supporting cell or a hair cell. And so once again, what we're doing in the Hearing Restoration Project is to look at normal and regenerating fish, normal and regenerating birds, and young and old mice to try and understand genetic pathways that may be used to produce new hair cells. And once again, in this process, we will be comparing young mice with older mice. And I just want to take a moment or two to underline again why we think this is important. Remember that a few moments ago I showed you that we can make many, many more hair cells in mice if we block the notch signaling pathway. However, we do this experiment in newborn mice. If we repeat the same experiment in mice that are just 6 days old, we see almost no new hair cells generated at all. So once again, it seems clear that as mammals get older, there are roadblocks being placed in the way of regeneration. In this case, roadblocks that interfere with not the notch signaling pathway. And what we're currently trying to understand in the consortium is what is the basis for these age-dependent changes that appear to block the regeneration process. So the take-home message that I want to get across to you today is that if we look at how birds and frogs and fish regenerate their hair cells and compare this to the absence of regeneration in mammals, it's clear that there are multiple roadblocks that prevent the mammalian cochlea from regenerating, as is seen in birds or fish. And so our challenge in the consortium for the next 5 years is to understand which genetic pathways are hit by these roadblocks in mammals, and to understand the nature of these roadblocks and how they work and how they may be removed. So in the final part of the talk, I want to tell you about another aspect of the work that the Hearing Restoration Project is looking at the moment, which is what happens to the cochlea after hair cell damage. What I'm doing here is showing you 4 different post-mortem samples of human cochlears. And these are from 4 different patients, all of whom had different degrees of hearing loss. And what I hope you can appreciate is that in these 4 different cases, the cochlea looks very different. On the upper left, it looks reasonably like a normal human cochlea. But as you can see, if you look in the other panels, in those patients, very few of the cells are remaining behind, and in their place are just simple flat cells that we believe are unable to participate in hearing. What we would like to understand is whether there are any supporting cells at all that are left here. The important thing here is that if we are to get hair cell regeneration from supporting cells, it's important that there are supporting cells remaining behind after damage. And so 3 members of the consortium, Albert Edge, Stefan Heller, and Elizabeth Esterly, are looking to see what happens to the organ of Corti after damage to see whether the supporting cells are remaining behind. And they are doing this using a technique called cell tracking. So I'm going to illustrate this to you in a simple series of cartoons. In the upper left panel, I'm showing you a cartoon of the organ of Corti with hair cells in yellow and their surrounding supporting cells. After hair cell damage, it is possible that the supporting cells die. If they die, they will be replaced by these more simple cells on either side of them. But this would be very important with respect to hearing restoration therapies, because if you design a therapy based on stimulating supporting cells to divide and turn into hair cells, it's obviously not going to be very successful if the supporting cells are already gone. On the other hand, it's possible that supporting cells don't die after hair cell loss, but they rather undergo an identity change and become more simple and nevertheless reside in the organ of Corti. And this would be important because if they remain behind in the organ of Corti, it is possible that we may be able to devise a way of coaxing them back into turning into hair cells with a view to regeneration. And so in order to ask this question, do supporting cells die or remain behind after damage? We're using techniques of cell tracking, and the way we do this can be illustrated quite simply here. What we will be doing in the Hearing Restoration Project is using genetic engineering technology to permanently label supporting cells, to give them a permanent genetic label, which I'm illustrating here with a simple blue color. So after hair cells die, the supporting cells will be left behind, and you can see them in blue in the upper panels. If the supporting cells also die, then when we come back in an older animal, we would not expect to see any of this genetic label left behind. If, on the other hand, supporting cells don't die, but rather just undergo some sort of transformation and become more simple, we should be able to identify these cells by the fact that they have retained this permanent genetic label. And so members of the Hearing Restoration Project are in the process of doing this experiment right now as a complement to our other studies to try and understand what happens to supporting cells after hair cell damage. And this is an important and complementary second part of our 5-year research plan. So in the last half hour or so, I've described to you the purpose and some of the strategies that we will be pursuing in the Hearing Restoration Project Consortium. What I'm doing here is showing you the members of the consortium. These are scientists in institutions all over the United States, and we have been going now for almost a year. We've been sharing ideas, sharing information, and I think this is really the true meaning of consortium. We are sharing and we are partners working together to address some of the problems that I've outlined to you in this talk. We're already exchanging information. In addition to having conference calls and periodic meetings several times a year, we are now online. We have our own website where the scientist members of the consortium can share ideas on a weekly basis. And we hope by working together we will be able to address some of the problems that I've been describing to you during this presentation. So thank you for listening to this presentation. I hope you've found it informative. And if you would like to know more about the Hearing Restoration Project, please go to the Hearing Health Foundation's website at hearinghealthfoundation.org.